So hello everyone. Today we are going to see the what are the important contribution in the development of microbiology. So scientists which is involved in microbiology. Okay. So there are various scientists it is involved actually. Okay. And these scientists play very important role in regarding to development of microbiology. So first one, that is Robert Hooke. He first time observed the microorganism, and he observed a plant material actually. Okay, and this plant material which was composed of little boxes, and he introduced term as an cell. So Hooke's observation were the ground work for the development of cell theory and the concept that all living things are composed of cell. So Robert Hooke, he having a year that is the 1635 to 1703, he prepared a compound microscope with a magnification power about 200, and he mainly studied the object of dry state, and he reported his finding to the Royal Society of London in 1665, in which he reported that the picture of mold but not in bacteria. Afterwards, before that, one scientist which is present which is in 1519, that is the Zernesen Jensen. This Zernesen Jensen, he simply magnify the glasses. Okay, and these glasses, magnifying glasses, it is used for the observation. He first time reported that using a second lens we can enlarge the image formed by primary hand lens. So we know that in microscope there are two lenses it is used. One is it is a objective lens, and another one it is a eyepiece. So he first time reported that when we are using a second lens, it can magnify the further. Okay, and he introduced a microscope with a 5 to 10x magnification power. So in our microscope, the objective lens, it is having a magnification of 10x, 45x, and the 100x, and so on. Okay, and this is a principle using two lenses. It is used in a compound microscope. So in compound microscope there are two lenses are used, and these two lenses are magnify, or they further magnify, or they produce a large image. So this is about the in regarding to the microscopy. Three scientists it is involved, that is Zernesen Jensen, afterward that is Robert Hooke, and next one that is Antony von Leeuwenhoek. And Antony von Leeuwenhoek, using a simple microscope, was first used to observe the microorganism. Okay, and the world of microbes was discovered with the help of simple microscope by the Leeuwenhoek, and he is a Dutch scientist actually. And this Antony von Leeuwenhoek was having a hobby of lenses grinding and the microscope making, and through that hobby he observed a microorganism. So it is a very important contribution that the first time the microorganism it is observed by the Antony von Leeuwenhoek. Okay, so these are the three scientists who involved in microscopy. Afterwards, there is very important contribution of Louis Pasteur. He disproved the SG theory. SG means that is the spontaneous generation theory. Okay. This is what a very short highlight. This short highlight we first see, and afterwards we are seeing in a brief. Okay. So first one, he disapproved the SG theory, means he opposed the spontaneous generation theory. Okay, and he discovered the fermenting fruit to the alcohol by the microbes. From now the fermentation was started, and he saw fermentation started and sorted with a different microorganism, giving a different taste of wine. Okay, so he first time see that different types of organism it is involved, and these different types of organism it is giving a a different type of taste to the wine. Okay, then he selected a particular strain, that is the yeast, which is having a high quality of wine. So the strain yeast, it is responsible for producing a alcohol fermentation, or this yeast, it is nothing but a Saccharomyces cerevisiae. This Saccharomyces cerevisiae, it is giving a high yield of that wine. Okay, then he developed a method to remove the undesired microorganism from the juice without affecting its quality. Okay, so he developed a method like it remove the undesired microorganism from the juice. Okay, without affecting quality. So how we can remove the undesired microbes? We can remove the undesired microbes like heating the juice at 62.8 degrees Celsius for half an hour did the job. Means what? You have to take a simply the juice. When we are giving a heating up to a 62.8 degrees Celsius, means approximately the 63 degrees Celsius, it can remove undesired microbes. Okay, and it is carried out for half an hour. Okay, so this technique he called as an pasteurization, and this pasteurization technique, it is commonly used in the field of milk industry. Nowadays when we see any milk bag, on that milk bag there is a written like a pasteurized milk. Okay, so this is very important process of pasteurization, which can remove the undesired microbes from that content. Okay, then he discovered the parasite. He discovered one parasite, that is the protozoa, and this protozoa, it is having, or it is causing a pip-brine disease of a silkworm, and he suggested that the disease free caterpillar can eliminate the disease. Okay, and he isolated the anthrax. Okay, so he isolated the anthrax organism. This anthrax, it is this anthrax disease, it is set up due to the bacilli, and this anthrax, it is found this anthrax, it is caused by the bacillus anthracis, which is a bacillus one. Okay, and this bacillus, he taken, or he identified from the blood of cattle, sheep and the human being. Okay, then he also demonstrated the virulence, that is the ability of microbes to cause a disease. Okay, so he demonstrated one concept known as a virulence, and virulence, it is nothing but the ability of that microorganism which cause a disease. Okay, and he developed a vaccine, that is the attenuated vaccine. This vaccine, it is used against the rabbit from the rabies, from brain and the spinal cord of rabbit. Okay, so now we see the one by one what are the main contribution of the Louis Pasteur. Okay, so this Louis Pasteur, he disapproved the spontaneous generation, means he opposed the spontaneous generation. Means what do you mean by the spontaneous generation? Spontaneous generation, it is a state that the life could arise spontaneously from inanimate object or inanimate material, that is the non-living thing. Okay, so the non-living thing means the spontaneous generation here, it is spontaneous generation, it is called as abiogenesis. Abiogenesis means what? The living thing, it is arises from non-living thing, it is known to be as an spontaneous generation. Okay, so after Leeuwenhoek had revealed the vast number of microorganisms which is present in nature, and scientists begin to wonder about their origin. Okay, so from the beginning there were two thoughts. Some believe that they were from the seeds or germs. Some believe that it is came from non-animate objects. Okay, and the belief in the spontaneous formation of living being from non-living matter is called as a spontaneous generation, or it is known as abiogenesis. So spontaneous generation, it is known as an abiogenesis. So Pasteur, he approved the theory of spontaneous generation. He strongly supported the theory of biogenesis. So what do you mean by the biogenesis? Biogenesis means what? The life, it is originate from the pre-existing life form. So life which is originate from a pre-existing life form, it is known to be as an biogenesis. Okay, and to prove this, he carried out a several experiment. Pasteur poured the meat infusion into the flask and then drew the top of each flask into a long curved neck that could admit air but not a dust. Means what? He make a goose hook neck, okay, and he took out the meat infusion. मीन्स एक असा फ्लास्क घेतला की ज्याला नाही एक गूस नेक म्हणजे असा मोठा नेक असलेली एक फ्लास्क होता. त्या फ्लास्कमध्ये ही टेक आउट द मीट इन्फ्युजन अँड दिस मीट इन्फ्युजन, तो फ्लास्क असा डिझाईन होता की इट कुड नॉट ॲडमिट द एअर, ओके, और इट कुड नॉट ॲडमिट द डस्ट. ओके, सो मग एअर पण येणार नाही आणि डस्ट पण येणार नाही, ओनली द मीट इन्फ्युजन प्रेझेंट असेल. अँड दिस मीट इन्फ्युजन ही हीटेड. So he found that if infusion were heated, they remain sterile until they are exposed to the dust. Okay, means simply he took out the special flask which is called as a goose hook neck flask. And this goose hook neck flask, from this goose hook neck flask, he designed such a way that air it is not coming inside, and the dust it is not coming inside, because we know that air contain a lots of microorganism. So after opening them on a dusty road or and resealing them, he demonstrated that the growth of microorganism in all the flasks. So unopened flasks were sterile, whereas open flask it become unsterile. Unsterile means what? Unsterile means where there is a microorganism it is found. Thus he approved the theory of spontaneous generation. So he say that the living thing it is arises only from living thing, not from non-living thing. Okay, so this is a very important contribution of a Louis Pasteur, that saying that he carried out one experiment like a goose hook neck experiment, in that he took out a meat infusion, he heated them. So during heating what happened, the microorganism it get killed. So this heated infusion it is remain as a sterile one. But when this goose neck it is reopened, at that time the air it is come, dust it is came, along that air and the dust the microorganism it is came, microorganism it is grow there, and the flask become a unsterile. So this is very important contribution of a Pasteur, who disapproved the spontaneous generation. Then the role of fermentation. Okay, so role of fermentation means what? That is the Louis Pasteur first time said that fermentation is a biological process. So who first time say that the fermentation is a biological process? It is by the Louis Pasteur. Before him, according to the one scientist known as von Liebig, von Liebig, VON, okay, LIEBIG. So this von Liebig, he say that fermentation it is an purely a chemical process. Okay, so Pasteur found that alcohol fermentation was brought about by the microbes. Okay, he observed that different types of bacteria in different lots. In good lots, one type of organism it is a predominant one, while in poor product another kind of organism was demonstrated. So Louis Pasteur he discovered the alcohol production. This alcohol production it is carried out with the help of grape juice, and it was due to the yeast, that is Saccharomyces cerevisiae. So the presence or the contamination of rod-shaped bacteria resulted in the large amount of lactic acid production in a wine. So we again in next bit we see in more detail about the what is the role of fermentation, what is the role of microbes in fermentation. In that see this Louis Pasteur he when he tested the grape juice, he found one type of bacteria, that is the rod shaped bacteria. This rod shaped bacteria, it is nothing but a lactobacillus bacteria, and this lactobacillus bacteria, it is having ability to produce a lactic acid. So he found that microorganism in fermented fruit or grain result into the alcohol production, and he coined the term fermentation. Okay, so this is very important contribution. While he studying the fermentation, he also discovered one important contribution, that is the he discovered the anaerobic life. Because whatever the fermentation process, it is carried out in absence of oxygen, and where there is oxygen, it is absent, it is the condition, it is called to be as an anaerobic. Okay, so Louis Pasteur first time reported the presence of anaerobic life. Okay, while studying a sample from a butyric acid fermentation under the microscope, he observed that organisms in a drop of sample were active at a center but non-motile at a periphery of a drop. So he concluded that the organisms are favored in absence of oxygen, and he devised the term anaerobic microorganism. सो जेव्हा लुईस पॅश्चर ब्युटेरिक ॲसिड फर्मेंटेशनची स्टडी करत होता, तेव्हा त्यांनी एक हँगिंग ड्रॉप टेक्निक असते, ज्यामध्ये तुम्हाला कळतं की मोटिलिटी कशी असते. ओके, सो त्यामध्ये त्याला कळालं की जेव्हा नाही आहे का ऑक्सिजन येत आहे, at that time the organism means at a periphery of that drop, the organism become non-motile, means in absence of oxygen the organism it become non-motile. Okay, so first time he discovered the anaerobic life. Okay, afterwards the very important contribution, it is a pasteurization. Okay, so Pasteur observed that preservation of food material can be prolonged by heating